Everyone, welcome to biology class. In this class, we shall see about 11th standard botany unit 6, the cell. In this part of the video, we will discuss about the special types of chromosomes and about flagella and cilia. First, special types of chromosome. In the last class, we have seen about the different types of chromosomes and its types, the types of chromosome based upon the position of centromere and based upon the functions. So this class we shall discuss about the special types of chromosomes. So what are special types of chromosomes? These chromosomes are larger in size and hence they are referred to as giant chromosomes. And these kind of giant chromosomes are found in certain plants and they are also found in the suspensors of the embryo. The suspensors are nothing but the supporting tissues of the embryo. And examples for these such giant chromosomes are number 1, Polyteen chromosome, and number 2, Lambrush chromosomes. First, we shall see what are Polyteen chromosomes. Polyteen chromosome. They are first observed in the salivary gland of Drosophila. Drosophila is nothing but a fruit fly, and this was observed by E.G. Balbiani in 1881. This kind of chromosome was first observed in Drosophila by Balbiani. And in larvae of many flies, midges are also a kind of insect, and some insects, the interphase chromosomes duplicate. So during the interphase, the cell will prepare itself for the division. So the DNA will start, the chromosomes will start to duplicate, and again they are going to reduplicate. So there is duplication occurs without any nuclear division. So as a result of that, the chromosome will contain multiple copies of structure, okay, because they are not going to divide, they are just going to duplicate. So there will be multiple copies of chromosomes found, and such a chromosome is referred to as Polyteen chromosome. So many chromosomes, they are joined together without any nuclear division, and this is seen in light microscope itself. So usually the chromosome structure will not be visible in a normal light microscope, but this Polyteen chromosome, because of its giant structure, it can be seen in light microscope also, and they are generally genetically active. And this is the picture which is representing the Polyteen chromosome, and you could see there is an alternating dark band and light band. So you could see dark band, and alternating to the dark band you have a light band which is referred to as interband. And about 95% of DNA are present in bands and 5% is found in the interband. And the Polyteen chromosome here it has got some extremely large puffed region, and this region is referred to as Balbiani ring, or otherwise referred to as chromosome puff. Okay, so this structure, extremely large puff structure, this is referred to as Balbiani ring or chromosome puff. This kind of puff in the Polyteen chromosome is seen in Chironomus larvae. It is also a kind of insect larvae. Okay, they belong to the category of flies called midges. So in that also you can find this Balbiani ring. And puffing of bands are the sites of, so why this puffed region is found? So they are nothing but the sites of intense RNA synthesis. So these regions, more RNA is synthesized, so it is represented as a puffed region. And this chromosome, this Polyteen chromosome, it is also found to occur in the salivary gland, and hence this chromosome is also known as salivary gland chromosome. So instead of they are asking as a Polyteen chromosome, even they can ask us write a note on salivary gland chromosome, so which is nothing but Polyteen chromosome. The gene expression, the transcription of genes and RNA synthesis occurs in the bands along the Polyteen chromosome. So all these processes like gene expression, transcription and RNA synthesis will occur in the bands of this Polyteen chromosome. The next type of giant chromosome is Lambrush chromosome. So this Lambrush chromosome, they occur at the diplotin stage of first meiotic prophase. In prophase you have several divisions. leptotene, zygotene, pacatene, diplotin, and diakinesis. So this Lambrush chromosome, they occur at the diplotin stage of the first meiotic prophase, and where it is found? It is found in the oocyte of an animal called salamander. So in salamander also you could find this Lambrush chromosome, and apart from that also it is seen in the giant nucleus of the unicellular algae, okay, which is acetabularia. So in this algae also this Lambrush chromosome is found. And who first observed this Lambrush chromosome? It was first observed by Fleming in the year 1882. This Lambrush chromosome is a highly condensed chromosome. Okay, so it is a condensed chromosome. This condensed chromosome is going to form the chromosomal axis. So from which, so this is the chromosomal axis. From the chromosomal axis arise the lateral loops. Okay, so lateral. So here you can find the lateral loops. This lateral loops of DNA extend as a result of intense RNA synthesis. How you, in case of Polyteen chromosome, we have seen chromosomal puff, okay, which is formed as a region of intense RNA synthesis. Similarly, in Lambrush chromosome, so this lateral loops arise as a result of intense RNA synthesis. So these are the special types of chromosome. The special type of chromosomes is otherwise referred to as giant chromosome, and Polyteen chromosome and Lambrush chromosomes are the examples for giant chromosome. And Polyteen chromosome is first observed by Balbiani in the salivary glands of Drosophila. Hence, it is also referred to as salivary gland chromosome. So, this is the Polyteen. And next, we have seen about the Lambrush chromosome. It is observed in salamander and a unicellular algae called acetabularia. And this is the structure of the Lambrush chromosome. The next topic we are going to see is about flagella. So flagella, so even flagella are found in both the prokaryotic organisms and also in eukaryotic organisms. So first we will discuss about the flagella in case of prokaryote. So it is referred to as prokaryotic flagellum. Example for a prokaryote is bacteria. So bacteria, certain bacteria are found to have this flagella. So flagella are nothing but the helical appendages, and this flagella are helpful for motility. Motility is nothing but movement. Okay, so they are helpful for the movement of the bacteria, and they are much thinner than the flagella or cilia of the eukaryote. The prokaryotic flagella is very thin when compared to the eukaryotic flagella or cilia. And the flagella will contain the filament which will contain a protein called flagellin, and the structure consists of a basal body associated with the cytoplasmic membrane. So this is the basal body. So the flagella is anchored in the basal body. So this basal body is associated with the cytoplasmic membrane and the cell wall. And with also apart from the basal body, it has got a short hook-like structure, and also it has got a helical filament. Okay, so it consists of three parts. Number one, basal body. Number two, hook. Number three, filament. So these are the three parts of a prokaryotic flagella. And bacteria will rotate their helical flagella and propel the rings present in the basal body. So here you could see, these are the rings. So these rings are found in the basal body. So it is going to exhibit a rotatory movement, the rings. So when the rings are going to exhibit a rotatory movement, the flagella which is anchored also will move. Okay, so thereby it will create a force which will drive the bacteria to move. So bacteria, there are two kinds, gram-positive bacteria and gram-negative bacteria. So already we know. In gram-positive bacteria, only it has got two basal rings. So this is a gram-negative bacteria. Whereas in case of gram-negative bacteria, only two basal rings are seen. One is S ring and the other one is M ring. S ring is attached to the peptidoglycan. Peptidoglycan is nothing but a constituent of the cell wall. So S ring is attached to peptidoglycan, M ring is attached to the cell membrane. So in case of gram-positive bacteria, only two basal rings are found. Whereas in gram-negative bacteria, two pairs of rings are found. One in the proximal end and the other one at the distal end. Okay, so both are connected by means of a central rod-like structure. So 4 different kinds of ring: L ring, lipopolysaccharide ring; P ring, peptidoglycan ring; S, supermembrane ring; and M, membrane ring. So these are the 4 different kinds. So here you could see L, okay, which is found in the cell wall region. Okay, so L ring, lipopolysaccharide ring; next P ring is the peptidoglycan ring; and S and M ring, supermembrane ring and membrane ring. So these are the 4 rings which are found in case of gram negative bacteria, whereas in gram positive bacteria only 2 rings are found. One is S ring and the other one is M ring. Now let's see the mechanism of flagellar movement. So in case of prokaryote, the flagellar movement takes place by means of proton motive force. So it is based upon the hydrogen ion or the proton gradient. The flagella will move. In flagellar rotation, only proton movements are involved. So usually ATP will be involved in kind of many activities of the cell, but in case of this flagellar movement, ATP is not involved. So which gives the force or the drive for the flagella to move is nothing but the proton. The protons flowing back into the cell through the basal body rings of each flagellum. So the proton is going to move through the basal body rings, and this proton is going to make the flagellum to rotate. And these rings constitute the rotary motor, and the proton motive force is nothing but the force derived from the electric potential and hydrogen ion gradient across the cytoplasmic membrane. So this is going to drive the flagellar motor. And for the rotation of flagellum, the energy is derived from the proton gradient across the plasma membrane generated by oxidative phosphorylation. And in bacteria, the flagellar motor is located in the plasma membrane, and in the plasma membrane only oxidative phosphorylation process. So phosphorylation is nothing but the production of the phosphate ion. The process takes place, and therefore the plasma membrane is a site of generation of proton motive force. Eukaryotic flagellum, cell motility. Now let's see the structure of the eukaryotic flagella. The eukaryotic flagella is enclosed by a unit membrane, and this also arises from the basal body similar to that of the prokaryotic flagella. The eukaryotic flagella is composed of outer 9 pairs of microtubules. So here you could see, so this structure is referred to as microtubules. So outer, how many microtubules are found? 9 microtubules are found, and in the center you have 2 microtubules. So outer 9 pair, okay, outer 9 pairs of microtubules, and in center you have 2, okay, not 2 pair, it is found 2 microtubules in the center. This arrangement is called as 9 plus 2 arrangement. So the flagella are microtubule projection of the plasma membrane, and flagella is longer than the cilium. And the structure of the flagella, it has got an axoneme, that is the basal shaft, okay, structure called axoneme, and it is made up of this microtubule, and which is constituting of a protein called tubulin. Movement. So how it is going to move? The outer microtubule doublet, okay, that is 9 doublet, is associated with the axonemal dynein, and this is going to generate the force for the movement. This dynein arm is going to generate the force for the movement. This movement is ATP driven. Okay, so in case of prokaryotic, it is proton motive force is involved in the movement, whereas in case of this eukaryotic, the movement which is driven by the energy in the form of ATP. The interaction between the tubulin, so this is the tubulin and this is the dynein. So this interaction is essential for the contraction of the cilia and flagella, and the dynein molecules uses energy from ATP to shift the adjacent microtubule. So this molecule it uses energy, and this energy it is going to shift the adjacent microtubules, and this movement bends the cilia or the flagella. And the last part of the lesson is cilia. So cilia, it is in plural it is referred to as cilia, and in singular it is referred to as cilium. They are the short cellular numerous microtubule bound projection, okay, so of the plasma membrane, and Cilium is a membrane bound structure and it is also similar to that of the flagella. It is also made up of basal body, rootlets, basal plate and shaft. And here the shaft, otherwise referred to as axoneme. Here also you have 9 pairs of microtubule doublets arranged in a circle along the periphery. So outer you have 9 microtubule pairs. Okay, and in center you have 2. Same arrangement that of flagella, eukaryotic flagella. And the microtubules here also it is made up of tubulin. And the motor protein dynein connects the outer microtubule pair and links them to the central pair. So it is going to connect the outer microtubule pair, it is connected, and also it is going to link it to the central microtubule by means of this dynein. And nexin, it is going to link the peripheral doublet. Okay, so the peripheral, the outer 9 doublets are there, no, they are linked by means of nexin. Okay, so that's it with the cilia. So in this part of the video we have seen in detail about the special types of chromosomes or the giant chromosomes called Polyteen and Lambrush chromosome, and also we have seen about prokaryotic and eukaryotic flagella and also about cilia. Meet you in the next session. Thank you.